So, welcome to all of you in today's session where we are going to learn about the acquisition of nutrients in plant. But before going into the detailed mechanism of solute transport, we must be aware of the structure and composition of biological membranes. So, you see that the biological membranes we will discuss in detail, but these are the contents we are going into detail. Uh, so, you see that we have gone through already movement of water molecules from soil to root and then uh, upper part of the plant. So, basically movement of solute and transport from soil solution to various tissues is one of the first process that required during the germination of seeds in the soil. And generally you see that seeds are usually very dry and containing less than 10% water. So, but when they start to germinate, they absorb water, and generally this water hydrate their cells and tissues. Apart from, you see that oxygen and carbon obtained from the air, plants generally get their water and nutrients from the soil through their root system. The movement of water molecules and ions from one place to another within a plant is generally called transport. When these solutes moved over long distance, it is basically called translocation. So, there is a subtle difference between transport and translocation. So, this intercellular distribution of water, ions and organic molecule is very crucial for plant growth, signaling between cells, nutrients and maintaining a stable internal environment that water, ions and organic molecules are distributed within the plant cell and between them. So, to achieve this important functions, plants generally have developed different transport mechanisms through basically that accompanied through two main distinct pathways. One is apoplast, the space between the cells, and the symplast, the interconnected network of plant cell, which we have already discussed in our earlier lectures. Now, you see that the interior of plant cell is generally separated from plant cell wall, and the environment by a sort of boundary known as plasma membrane. So, generally the lipid bilayer structure of plasma membrane repels water, keeping generally hydrophilic compounds like most metabolites either inside the cell organelle or within the cytosol. So, generally in addition to forming a this plasma membrane, in addition to forming a sort of hydrophobic barrier to diffusion, the membrane must facilitate the continuously and regulate inward and outward traffic of selected molecules and ions that generally cell take up various nutrients and export solute and thereby regulate its turgor pressure. So, basically in this chapter we will study about the detailed structure composition of plasma membrane one by one. So, you see in the slide that plasma membrane is very dynamic and fluid structure generally that forms the outer boundary of the cell, and plasma membrane basically functions as a selectively permeable barriers controlling the passage of molecules across the cell boundary. Selective permeability means that certain solute can cross the membrane more readily than others. So, this is known as selective permeability. So, various models have been proposed to describe this movement of solutes and basically the structure and composition of plasma membrane was revealed by a very well-known model known as fluid mosaic model by S.J. Singer and G.L. Nicholson in the year 1972. And this model proposed by Singer and Nicholson is widely accepted throughout the world. According to this model, the plasma membrane is generally depicted as mosaic of components. You see in the slide as a mosaic of components including phospholipids, cholesterol, proteins and carbohydrates, imparting generally a fluid nature of this membrane. And consequently, you see that membranes are basically conceptualized as a quasi-fluid structure in which proteins are embedded within this lipid bilayer. So basically, this fluid mosaic model reveals both the mosaic-like distribution of proteins dispersed toward the lipid bilayer and the fluid movement of lipids and proteins within the membrane. So now question arises, what is the basic composition of biological membranes or plasma membrane? So the basic principal component of plasma membrane that works together to form a selectively permeable barrier surrounding the cell are basically lipids, and there are two types of lipids, phospholipids and cholesterol. Then in addition to lipids, proteins and carbohydrate that are attached to some of the lipids and proteins. Now we will discuss in detail the detailed components of the plasma membrane. So first is lipid bilayer. So the basic structure of the plasma membrane is called lipid bilayer, and it's like a sandwich model of two layers of amphipathic molecules called lipids. These lipids generally possess both hydrophilic and hydrophobic region, and you might be aware hydrophilic water loving and hydrophobic water repelling regions. So especially their polar head groups of lipids interact with the intra or extracellular aqueous environment, while their non-polar tails orient inward, creating the hydrophobic interior of the membrane. The main driving force behind the organization of lipid bilayer are basically hydrophobic interactions, and you see that there are basically three main classes of lipid molecules present inside the plasma membrane, namely phospholipids, glycolipids and sterols. So we will discuss one by one. Now what is the structure of phospholipid? So basically you see in the slide that basically phospholipids, there are made up of four components, an alcohol like glycerol or sphingosine, then fatty acid, you see fatty acid, and another alcohol attached to the phosphate, this one. The fatty parts, the fatty acid part are generally hydrophobic, while the rest of the molecules are hydrophilic. You see this part provides hydrophobic nature of lipid bilayer, and this one is hydrophilic. And basically there are two types of phospholipids, namely glycerophospholipids and sphingospholipids, that are main phospholipids. Now in addition to phospholipids, glycero and sphingolipids we will discuss in detail. So glycerophospholipids, they are also called as phosphoglycerides, basically derived from glycerol and have fatty acid, phosphate and alcohol, for example choline. Different kinds of basically alcohol attached to the phosphate group that give different names to these molecules. For example, if you see in the choline, it is called phosphatidyl choline, also known as lecithin. So if serine is present, it is called phosphatidyl serine. Basically phosphoglycerides are the most common phospholipids present in the cell membrane or plasma membrane. So sphingospholipids have different backbone called sphingosine instead of glycerol that is present in phospholipids. They have these sphingospholipids also have fatty acids, phosphate and and alcohol attached to the phosphate. In sphingophospholipids, the amino group of the sphingosine is generally, you see, is linked to a fatty acid here by a special bond called amide bond, and the most common sphingophospholipid is sphingomyelin. Now, another compound is sterol. Sterols are basically known as steroid alcohols. Basically, they are very important class of organic compounds. So, the basic structure of steroid nucleus is made up of 4 fused ring, see in the figure, 3 with and 6 carbons and 1 with 5. So, one of the sterol is cholesterol, which is present in the plasma membrane of only animal cells, which acts as a temperature buffer in plasma membrane, but this cholesterol is generally absent in prokaryotes and plant. So, plant generally contains other sterols like campestrol, cytosterol and stigmasterol. In addition to this, you see that ergosterol is also type of sterol present in the cell membrane of fungi. So, you must note this important point that ergosterol is present in the plasma membrane of fungus. Now, next important constituent of plasma membrane are carbohydrates. So, these carbohydrates form the third major important component of the plasma membrane after phospholipids and proteins. So, you see in general they are found on the outside surface of the cell and bound either to proteins, glycoprotein or to lipids. Once they bound to lipid, they are known as glycolipids. And these carbohydrates contain carbohydrate chain may consist of 2 to 60 monosaccharide units and can be either straight or branch. You see how this glycoproteins and glycolipids, they are attached to the lipid bilayer. Now, membrane proteins are very crucial component of the plasma membrane, and these proteins can be generally categorized as peripheral proteins. You see, peripheral protein, then integral protein or lipid-linked proteins. So, basically these peripheral proteins are located on the surface of the membrane. You see how these peripheral proteins, they are located on the surface of the plasma membrane. And but very interesting aspect I would like to share that these peripheral proteins can be removed from the membrane using mild extraction methods such as exposure to solutions with high or low ionic strength or extreme pH levels. And basically these peripheral proteins, they are attached to the membrane through electrostatic and hydrogen bond interactions, and generally they do not penetrate the hydrophobic core of the lipid bilayer. So, that is why they can be easily removed from the plasma membrane. And generally these peripheral membrane protein, generally they bind to the membrane indirectly via the interaction with the lipid part of the lipid bilayer. And you see that these peripheral proteins can be attached either on the inner or outer side of the plasma membrane, and respectively they are known as intrinsic or extrinsic proteins. So, most of the peripheral proteins generally they are soluble in water, and these examples of peripheral proteins can be found in membrane of red blood cells like spectrin. Now, next protein is integral proteins. You see, integral membrane proteins, also known as tunnel proteins. and they are basically, you see in the slide that you see that basically these proteins, they are basically embedded within the lipid bilayer. You see they are embedded in the lipid bilayer and cannot be easily removed using simple extraction method. Just I discuss about the peripheral membrane protein, they can be easily removed, but integral membrane protein cannot be removed easily by simple processes. And these integral membrane proteins are basically transmembrane proteins, means they have one or more segment that penetrate through the phospholipid bilayer. And these transmembrane proteins contain amino acid residues with hydrophobic side chains that interact with the fatty acid group of the membrane phospholipids, and then that secure proper anchoring the protein to the membrane. So all transmembrane proteins generally both have hydrophobic and hydrophilic regions. So these proteins are basically amphipathic in nature. Then the third protein, you see the lipid anchored proteins, they are attached to the membrane surface through lipid molecules. Therefore, membrane proteins are linked to lipid molecules. They are known as lipid-linked or lipid-anchored membrane proteins, and these proteins, they form covalent bond with lipids. So basically, these three proteins, they are present in the lipid bilayer. Now, you see this is the slide showing that the different ions and charged polar molecules, uncharged large polar molecules, uncharged small polar molecules and gases, how they can cross or they can not cross the plasma membrane. So this is very interesting to discuss here, that you see that the interior composition of the generally cell is maintained due to selective permeable nature of the plasma membrane. You see this is the inside and this one is the cytosol. This one is the cytosolic side, and you see that the membrane, this membrane basically acts as a sort of barrier preventing the free movement of molecules between the cytoplasm and external cellular environment. Here is the external cellular environment, ions, charged polar molecules, uncharged polar molecules and uncharged small polar molecules and gases are present. So whether they can cross this plasma membrane to cytosol, or whether they require assistance of something else to cross this. So you see that the plasma membrane basically acts as a sort of barrier. So generally most biological membranes, in the most of the biological membranes, it is difficult to pass through the protein free lipid bilayer. And generally, if I talk about non-polar molecules like oxygen, you see like oxygen, carbon dioxide, they can easily dissolve in lipid bilayer and then diffuse across the plasma membrane. So this arrow shows they can easily diffuse across the plasma membrane. Now similarly, uncharged polar molecules such as water and urea can diffuse across a lipid bilayer, but contrast to this, lipid bilayers are highly permeable, highly impermeable to charged molecules like potassium, you see potassium, magnesium, calcium and bicarbonates. So basically this plasma membrane is impermeable to these cations and bicarbonates. However, the plasma membrane, unlike protein-free lipid bilayer, permits the passage of ions and various charged or uncharged polar molecules like sugars and amino acid. So basically, specialized transport proteins facilitate the transfer of such solutes across the plasma membrane. Therefore, you see that transport across the plasma membrane could occur either through the lipid bilayer or the membrane transport system, via, you see that facilitated via diffusion, that could be protein mediated and basically differ from the simple diffusion. So, the transport across the plasma membrane basically could occur via two ways. One is passive transport and another one is active transport. So, you see that these cations, bicarbonates, calcium, magnesium, and they cannot cross plasma membrane without the aid of anything else. But you see that carbon dioxide, oxygen, water and urea uh, can cross the plasma membrane by diffusion. So, this process, this transport across the plasma membrane could occur by passive and active transport.